Here with relation to biotechnology, we have two important scientists called Boyer and Cohen. Now we will be looking at the historical breakthrough or what is the major discovery in biotechnology which has really started this off as a sort of a new interdisciplinary science and a sort of a wave. Because we seem to be riding on the wave of biotechnology in the 21st century. If you take, there have been two important scientists whose names are Boyer and Cohen. Novel breakthroughs in biotechnology, especially by Boyer and Cohen, primarily had been with their sort of a work in relation to a microorganism called Escherichia coli. We understand Escherichia coli is the most famous bacteria in the biosphere. It lives in the gut of human beings. Its genome has been thoroughly mapped. It is very easy to handle. It can easily be grown in the lab. It multiplies every 20 minutes. And Escherichia coli is the favorite organism, model organism to be used in genetics and microbiology as well as in biotechnology. Now, if you take the first scientist, if you take Boyer. Now, Boyer was a sort of a scientist who worked in Pennsylvania. He was born in a place where there are a lot of mines and also railroads. And because there were a lot of mines, maybe his research was mind-blowing. And because he worked on railroads, that's one of the reasons he chose the DNA. So, Boyer's first experiment was, he was trying to study which are all the sort of enzymes which are present in the bacteria, and he found out that in bacteria there are a group of enzymes which are natively present, which are naturally present in the bacteria, which are termed as restriction endonucleases. And he found out that these particular enzymes could cleave certain of the sort of a DNA of any sort of a pathogen or any sort of an alien sort of a DNA which comes inside the bacteria. For a moment, if we just pause, if we just look at the structure of the bacteria. We understand that restriction, if you take a bacteria consists of a native DNA, a circular double-stranded DNA, which is a normal DNA of the bacterial cell. So, whenever you take any bacterial cell, in bacterial cells there are free-floating autonomous extrachromosomal circular molecules which are termed as plasmids. Plasmids are circular elements. They are naturally present in a bacteria. So, it's just like God's gift to biotechnology has been the presence of bacteria in Escherichia coli. And these were discovered by Boyer. So, Boyer tried to study what are the restriction enzymes which are present in the bacteria and what do these restriction enzymes do. The restriction enzymes act like molecular scissors. They act like chemical knives. They go and cut any sort of a foreign DNA which comes inside the bacteria. The bacteria, in order to protect itself from invasion of a foreign DNA, has reinvented and it has relocated within itself restriction endonucleases, which are just like home enzymes, which are just like in-house enzymes, which are naturally built up within the sort of a bacteria. So, these enzymes primarily would go and cut or prevent the sort of an alien DNA from logging into the sort of a plasmid or into the sort of a bacteria. So, later on primarily these restriction enzymes are very important property of both. They are both cutting enzymes and they cut with lot of biological precision. All restriction enzymes show biological position. They exactly recognize which part of the DNA to cut. Then they cut the sort of an enzymes, and they are also termed as molecular knives. So this was the sort of a discovery of Boyan. When he found out there is the presence of restriction enzymes, he also found when the restriction enzyme, when it cuts the DNA, the DNA leaves behind sticky ends. Now, what exactly is the meaning of the term sticky ends? Supposing we have an enzyme. Supposing we have an enzyme, it cuts the sort of a DNA. When it cleaves or cuts the DNA, it leaves a sort of an overhang. This overhang is termed as a sticky end. So the overhang is just like a sort of a adhesive tape. So the adhesive tape can again anneal, it can again join. So the overhang with the sticky ends can again join and again make a sort of a circular molecule. So the most important discovery of Boyer was Escherichia coli, presence of restriction enzymes being very specific for the foreign DNA, cleaving at specific sites, formation of sticky ends can anneal and they go to form circular DNA back. So this was the discovery of Boyer. Next we come to the discovery of Cohen. Cohen found out that in bacteria there is the presence of specialized circular rings which are termed as plasmids. Cohen discovered plasmids. So what are plasmids? Plasmids are extrachromosomal DNA elements. They are circular. Where are they present? They are present within the bacteria. So they are resident circular DNA elements. They are natively present. What happens to the plasmids? The plasmids show independent replication. That is, a circular molecule will again go to form another circular molecule within the sort of a DNA, within the sort of a bacteria. So plasmids are just like bubbles. Many plasmids will keep on multiplying continuously. They can get formed maybe 20 at a time or even 2000 at a particular sort of a time. And Cohen found out the method of removing and engineering the plasmids into other cells. So in short, Cohen was working on plasmids. He knew how to cut the sort of a bacteria, remove the sort of a plasmids and paste the sort of plasmids into another bacteria, say from plasmid A, from bacteria A into plasmid B, into plasmid A again into bacteria B. So Cohen primarily knew how to remove a sort of a plasmid A from bacteria A and put it onto bacteria B. Now it so happened that the breakthrough in research and the basis for biotechnology started when both Boyer and Cohen worked together. So when they worked together, they were able to isolate the plasmid. They were able to isolate the plasmid, insert the plasmid, and they studied DNA splicing techniques. So they had worked on isolation, insertion, along with DNA splicing. So, they could exactly know how to take the sort of a recombinant DNA A recombined DNA is the joining of two DNA fragments. They were able to join two DNA fragments of desired configuration. So, the most important breakthrough in biotechnology started somewhere in the 1970s. In the mid of 1970s, Boyer and Cohen, who had the background of restriction enzymes as well as the plasmids, they both came together in different labs of Stanford University and they engineered a plasmid. They could artificially create a sort of a plasmid. They could know how to remove the sort of a restriction enzyme, and they engineered the formation of an artificial recombinant DNA. They could manipulate the sort of a DNA such that you could get a sort of a DNA which could code with a desired sort of a character. And all these characters and all these properties were studied in vitro, that is in the test tube or in the sort of a lab. So, they could insert the DNA into the bacterial cells and they could develop a sort of a genetically engineered or a recombinant DNA in the cells for the synthesis of new or novel proteins. So, they could genetically engineer as you look through, we have a recombinant DNA. The goal or the major focus of Cohen and Boyer was how do you get a recombinant DNA and how do you get a sort of DNA with a desired configuration. So, they could do in vitro studies, that is within the lab. They could exactly engineer a plasmid and a DNA and the restriction enzyme and get an artificial DNA. And this artificial DNA which is termed as genetically engineered DNA that could produce novel proteins. Novel proteins are new proteins which are hitherto not known. A protein would be it could be a component from a prokaryotic cell as well as an eukaryotic cell. It can have maybe the properties of a plant cell as well as an animal cell as well as a microbial cell. Thereby, biotechnology primarily aims at using basic principles of engineering, chemical engineering, microbiology, genetics, physics, maths, computers, and thereby truly proves to be a sort of an interdisciplinary frontier. It's an interdisciplinary frontier science of biology wherein you primarily manipulate cells, tissues, organs, molecules, and to develop newer sort of newer properties, and products and processes which are useful for mankind. Now, for a while, if you just look back, if you take biotechnology which has been practiced right from the times of Lord Krishna, like the churning of the butter, making of the buttermilk, and right to making of the milk and the curds, till the popular foods which are on the streets, like maybe a sort of a bel puri and a pani puri, or maybe the sort of a foods which you normally eat like idlis and dosas, are all fermented biotechnology, and they all come under the domain of old biotechnology. So, old biotechnology relates to manufacture of vinegar, manufacture of butter, manufacture of milk, and maybe old home technologies of making butter, cheese, etc. And also traditional Indian foods like like idli and dosa. However, today if you take biotechnology, today's biotechnology deals with genetically modified organisms. We are dealing with relation to cells, tissues and organs. We are looking at whole systems, or we are looking at living organisms. We are dealing with genetically modified organisms and their sort of a products which have been developed, like a novel protein which is developed is termed as a product. And we are looking in terms of biotechnology and its application to human beings. Thereby, we are looking in terms of application of techniques to human beings, like what could primarily come into your mind. You could be taking into consideration the property of insulin. Insulin is a very important sort of an artificially developed enzyme for the treatment of diabetes. If you want to make a sort of a scale of human insulin in the lab, it would take lots and lots of time. Whereas if you genetically engineer insulin, you could be getting large scale. On a large scale, we have the success rate of production of insulin. So biotechnology primarily aims at large scale applications to human welfare. Thereby, if you really want to understand or have a good knowledge of the basics and also the sort of a understanding of biotechnology, in today's world, biotechnology has applications in vitro fertilization, test tube fertilization of human beings, test tube fertilization in plants. It has also got lots and lots of applications in terms of gene synthesis. If you recollect, we had an Indian Nobel laureate Hargobind Khurana, who is famous for synthesis of the artificial gene in the test tube. Then we can also develop DNA based vaccines. DNA based vaccines, like you can also develop vaccines related to banana. Plant based vaccines which you can feed to babies and mothers. And the strongest application of biotechnology is in medicine with relation to gene therapy. If ever we want to understand about how do you treat genetically inherited diseases, newer diseases, how do you create a sort of a solution for newer vaccines, newer viruses like Ebola virus as well as AIDS virus, genetic engineering or biotechnology seems to be the sort of an answer. Now, exactly till now I was just trying to bridge the sort of a gap between old biotechnology as well as new biotechnology. Now, if you take the definition, European definition of European Federation of Biotechnology, they primarily define biotechnology in a different sort of a way. And this definition holds good, especially in terms of learning. In terms of learning, the EFB definition of biotechnology runs as follows. European Federation of Biotechnology, we are integrating natural sciences. That is, we look in terms of cells, we look in terms of organisms, we also look in terms of molecules or their molecular analogues which are synthesized, chemically synthesized in the lab. And we seek out what could be the sort of a products and services. Especially in terms of their uses to mankind. In terms of food, in terms of industry, in terms of medicine. So we are looking in terms of large scale production of biotechnology products. So we are looking in terms of upgradation. The European Federation of Biotechnology lists biotechnology not as a cosmetic science. It is not a decorative science to be done in the lab. Biotechnology need to come out. Biotechnology is aimed, the broadest aim of biotechnology is it should serve mankind. So according to the European Federation of Biotechnology, it says we need to integrate natural sciences, organisms, cells, molecules and molecular analogues in terms of their products and services for the benefit of mankind. It has got applications in food, in industry as well as in medicine. We are looking in terms of large scale application. and we are in terms of life essential processes with relation to quality of life. This is the core of biotechnology. So, people who are skeptic about biotechnology or people who feel that biotechnology is bad, the aims of biotechnology are very, very clear. We are looking in terms of enhancing the quality of life, enhancing essential life processes like survival, like growth, like maybe how do you prevent a sort of a blood clot, or how do you treat a sort of a cancer patient. How are we looking in terms of better longevity? How are we looking in terms of gene therapy? How are we looking in terms of molecular diagnosis? Molecular medicinal diagnosis plays a very important role and it is central. Thus, careers in medicine, careers in agriculture, careers in environmental sciences have all applications in biotechnology. It is not as if biotechnology is separated from plant breeding or crop improvement. Biotechnology is an integral part of plant breeding. Thereby, if we look in terms of the definition of EFB definition of biotechnology, we seem to be coming in terms of looking at a sort of a definition of biotechnology which is both old as well as new. The principle, now we are coming exactly after we finish, we have just started off with relation to biotechnology. Maybe you are understanding what is the concept of biotechnology, what is the definition of biotechnology. Now we are looking in terms of the principles of biotechnology. When we look at the principles of biotechnology, we understand biotechnology primarily means manipulation of the genetic material. It is not as if we have started manipulating the sort of genetic material as of now. Thousands of years back itself, when agriculture had started, we were in the process of manipulation. So, manipulation of the genetic material, that is the principles of biotechnology, as well as introduction of the manipulated DNA into the sort of host cells and change in the phenotype of the organisms. These are the three important basic principles of biotechnology. Now, we are primarily, after finishing the introduction to biotechnology, that is block 1, we are entering into block 2, that is what is the conceptual foundation for biotechnology, or what does biotechnology primarily depend on. When you take biotechnology primarily, it's very, very important that we should have a conceptual background. That is, we should exactly know what is the theoretical background for biotechnology. The theoretical or the conceptual background for biotechnology rests on two important principles. On one side, it is based on genetic engineering. On the other side, it is based on plant tissue culture. So, we are looking at cells, we are looking at molecules. We are trying to manipulate the cell or the molecule, which is primarily a chemical. So, we are using engineering principles of biophysics and biochemistry onto life sciences or onto life molecules. That is one of the key reasons why the science is termed as biotechnology. We are following technological process, technological principles and trying to manipulate cells, molecules and genes. And to understand this, we need to have a conceptual foundation. And the conceptual foundation is built on two important premises, which is genetic engineering as well as plant tissue culture. Now, what exactly happens in genetic engineering? In genetic engineering, we manipulate the genetic material. There is manipulation of the genetic material. That is, we take and we try to alter, we try to change, or we try to speed up, or we try to delete, or we try to duplicate. Otherwise, we try to change them, we try to manipulate, we try to put a foreign gene of interest into the sort of a native or the sort of a plant DNA. We take a gene from maybe a sort of a bacteria and try to load it into a sort of a plant DNA. We are trying to manipulate the genetic material. After we manipulate in the lab, we take the sort of foreign gene of interest and through a sort of a plasmid, which is just like a sort of a vector, and we try to put it inside a sort of a bacteria. We introduce it into the host cell. The host cell could be a bacterial cell, or the host cell could also be a sort of a plant cell again. A is we do manipulation, B we try to do is introduction. After we introduce, we change the phenotype, we change the morphological character of the molecule or the sort of a cell. So, steps which lead to A, manipulation, step number B which leads to introduction, and step number 3 wherein we can exactly see that we have a recombinant DNA or a recombinant cell. All these are 3 different stages of genetic engineering, which is part A of the conceptual foundation. Parallel to genetic engineering in biotechnology, it is also based on plant tissue culture. Now, what exactly is meant by plant tissue culture? The cultivation of isolated cells, tissues or organs or an explant like a root, stem, leaf, flower, anther or a pollen on a nutrient or on a well-defined medium under controlled conditions is termed as plant tissue culture. So, in plant tissue culture, what do we have? We are looking in terms of growth of the cell, growth of cells. We are looking in terms of multiplication of cells. We are looking in terms of culturing of the cells. How long it should be cultured, how long should a pollen be put in a sort of a medium and allowed to go to form the sort of a mature sort of a pollen grains. So, we try to grow, multiply and culture them on exactly on a defined medium, a specific medium, which is a very complex medium because a normal cell cannot just be growing on a culture medium. It does not have roots or stems or leaves, so thereby it requires essential elements like carbon, hydrogen, oxygen, nitrogen, sulfur and phosphorus. So, it should be grown on a defined medium which is very, very specific for that particular sort of an organ or specific for that particular cell, like an embryo, like an endosperm, like an anther, like a pollen, or maybe like a sort of a root system, or maybe a sort of vegetative leaf. There are different mediums which are specified for different sort of parts as well as for different sort of plants. So, it is grown on a defined medium, very importantly under sterile conditions, because if we grow them under non-sterile conditions, it can lead to contamination. So, all biotechnology labs are highly sterile. That is one of the reasons why biotechnology in biotechnology labs there is high degree of sterility and no one is permitted. And we work in a very, very highly sterile environment, because if there is a small contamination, it can affect the entire sort of products which are present in the sort of a pipeline. It also involves growing of the sort of a plant cells, tissues under controlled conditions. It's very, very important that we grow them under controlled conditions, that is controlled conditions of time, of temperature, light, relative humidity and water, and the manipulation is for a large scale. So, by integrating some basic principles of molecules called genetic engineering, as well as integrating some basic methods of plant tissue culture wherein you grow molecules, cells and tissues under defined medium, under sterile conditions, on a large scale, under artificial conditions and again under controlled conditions. When we integrate both of these, it is very easy to manipulate cells and tissues, and thereby the starting point for biotechnology can be achieved practically. Now, this is one very important sort of a link. So, biotechnology to start with requires that anybody, a learner-centered person in biotechnology knows the basic principles of genetic engineering, and you should know the basic principles of plant tissue culture. Now, how can one cell give rise to the entire plant? If you recollect your knowledge of plant anatomy, we understand that every cell has got the innate ability or the genetically inherent potential of every cell to go to form the entire organ. This is termed as totipotency. So, totipotency is the innate ability of a particular individual plant cell to give rise to the entire organism. So, primarily it is based on the basic properties of cells. And when you take any particular cell, a cell is a basic unit of life. It contains a sort of a DNA. It contains even the sort of a semi-permeable membrane. These are the two important components of all cells, and all cells arise from pre-existing cells. So, thereby an integrated knowledge of cell biology, genetics and biochemistry and even basics of anatomy and embryology is essential for plant biotechnology. Now, as a second part of the conceptual development in the case of biotechnology, we exactly want to know there are two important parts of this. Part 1 says, okay, we had sexual reproduction in plants. Sexual reproduction in plants gives rise to lots of variability. Then again, we have asexual reproduction in plants, which gives rise to lot of uniformity. So, what advantages does biotechnology have if ever we want to manipulate plants? The advantage of biotechnology is when you do conventional plant breeding, when we look at the conceptual foundations of biotechnology, we understand that when you take conventional plant breeding, in conventional plant breeding, that is when a pollen lands on the stigma of any particular plant and we get the flower and the fruit, it is always said that we get both desirable and undesirable genes and characters. So, we do not have a sort of a choice, it is just a chance. Many a time it would be like we would be getting a sort of a plant which is high yielding, but also highly disease susceptible, because it has got a disease susceptible gene along near to the high yielding gene. Thereby, in plant breeding, which has been successful from so many years, conventional plant breeding carries both desirable as well as undesirable genes. And in conventional plant breeding, we have got two important modes of reproduction. One is the sexual reproduction, and the second one is termed as asexual reproduction. So, in sexual reproduction, it is the basis for variation and for natural selection, which has been quite successful all through geological time and in biological scale. Whereas, when you take asexual reproduction always leads to uniformity. So, there is preservation of characters. So, both using sexual and asexual reproduction and going by the conventional plant breeding, somehow or the other, though we have desirable genes, we also have undesirable genes. Now, what advantage or what is the sort of a take for biotechnology here? When you take biotechnology or genetic engineering, which is the new science of today, biotechnology is an integral part of plant breeding. It comes under the broad umbrella of plant breeding. It is an integral part of plant breeding. Biotechnology is a science, and it is an interdisciplinary science wherein it aims at doing precision engineering. That is, exactly we can locate your sort of a gene of interest, we can manipulate your gene of interest, we can modify your gene of interest, we can introduce the gene of interest into a host cell like a bacteria or a plant, and again we can select from them, as well as we can also multiply it on a large scale and improve the quality of life. Like if you take genetically engineered rice, which is termed as golden rice, has biofortified vitamin A, that is vitamin A enriched rice is available for mankind. And again, if you take another sort of a variety, like if you take flavor saver tomato, flavor saver tomato is a genetically engineered tomato which primarily does not soften or does not collapse under very cold conditions. So, it has got improved shelf value, and thereby you can get more of pulp for tomato sauce, for tomato ketchup, and also the sort of food industry. Thereby, we have got here two instances wherein we have the success rate of genetic engineering. Same is the case with relation to insulin. If we want to isolate the insulin gene, it is very easily done by means of genetic engineering. So, genetic engineering or biotechnology can eliminate undesirable characters, and it can only take up the desired character, and we are looking at precision engineering. So, this is the second important conceptual foundation. Conceptual foundation number one said we need to have a good knowledge of genetic engineering and plant biotechnology. Concept number two says other than plant breeding and other than basic techniques of sexual reproduction and asexual reproduction, plant biotechnology offers itself as a sort of a new wave science, wherein we can exactly know how to engineer a particular gene, manipulate the gene, as well as produce it in large quantities in bacterial cells which can easily multiply. Thereby, we have the sort of a choice of a selection process. The choice of a selection process is very much important for advances in agriculture, science and technology and medicine in the 21st century. Now, we come to the sort of a third foundation or the conceptual foundation of biotechnology, which is the construction of the first artificial recombinant DNA molecule. Now, primarily we would be studying how do you really develop the first recombinant DNA molecule. We again go back to the experiments of Cohen and Boyer. Now, when you take Cohen and Boyer, they first constructed they are the first persons in 1972 to construct the first artificial recombinant molecule in the world. They were the first scientists to develop the first recombinant DNA molecule. So, biotechnology takes its roots in early 1970s. So, they had found out that they had identified a gene which codes for an antibiotic resistance in Salmonella typhimurium. They had found out a gene which codes for antibiotic resistance, and they had taken the gene and placed it into the plasmid of Salmonella typhimurium. The gene was exactly present in Salmonella typhimurium in this sort of a gene. By restriction enzymes, by using restriction enzymes which can cut the sort of a plasmid, they could engineer the plasmid, they could re-engineer the plasmid. That is, they could take the plasmid of Salmonella typhimurium, exactly put the gene of interest for antibiotic resistance, they could load it into the sort of a plasmid of Salmonella typhimurium. After loading it, they had used an enzyme called DNA ligase, which could stick the sort of a gene into the sort of a plasmid. And later on, they placed it inside Escherichia coli. So, you have the gene of interest. The gene of interest codes for antibiotic resistance. It has been put inside a sort of a plasmid of Salmonella typhimurium. It has been genetically engineered, and the gene is placed into the sort of a basic host which is termed as Escherichia coli. Now, Escherichia coli has got the re-engineered plasmid. After it has got the re-engineered plasmid, this bacteria which has got the sort of a molecule which is re-engineered, it has got a sort of a recombinant DNA molecule. So, we have the first development of the recombinant DNA molecule in Escherichia coli. So, when it is loaded into the Escherichia coli, the recombinant DNA molecule, because it is present in a plasmid, the DNA becomes circular, it is autonomous, that is, it is independent of the replication of the bacteria, and again it is capable of speed replication. It is capable of speed replication because it has one very important sort of a molecule which is termed as an origin of replication. The circle has got a sort of a start button which is called a sort of an origin of replication. Thereby, your recombinant DNA molecule in Escherichia coli, you can make multiple copies of the gene of interest. It is just like how you take a sort of a, you take multiple copies of a paper on a Xerox machine. Similarly, within the sort of a bacteria, if a sort of a plasmid has got a sort of an ORI, you can make multiple copies of the gene of interest within the sort of a bacteria itself, and this technique or this process is in simple terms called as gene cloning. So, when you do gene cloning, you maintain multiple copies of a gene of interest, and from there you can again transfer it. So, thereby what are the basic principles of the development of the first recombinant DNA molecule by Cohen and Boyer? Gene of interest, antibiotic resistance. Next, introduced into Salmonella typhimurium, and again the plasmid is loaded into E. coli, and E. coli has got the sort of recombinant DNA molecule. The recombinant DNA molecule is genetically engineered, and the recombinant DNA molecule has got an ORI, and because of the presence of the ORI in a plasmid, you can make multiple copies of your gene of interest, and making multiple copies of your gene of interest is termed as gene cloning. So, thereby we understand whenever we are dealing with relation to biotechnology or plant genetic engineering, when you say genetic engineering or when you are looking in terms of genetic modification, there are some basic principles, or you can call them as the steps in genetic modification. The steps in genetic modification are: you identify the gene of interest, that is one. Your gene coding for antibiotic resistance. Next, you introduce the identified gene into host cells. You are introducing the identified into the host cells, that is the bacteria. Third, you maintain the introduced gene in the host cells. You also maintain it for some time, so that it becomes a sort of a resident gene, so that it is maintained in the sort of a bacterial cells. And later on, from there, it can be transferred to the progeny of Escherichia coli. As you understand, E. coli can multiply every 20 minutes, and thereby you can get millions and millions of copies of your gene. Now, if the same sort of an experiment is done, imagine a sort of a lab as big as a small room, and imagine the sort of a lab to be totally packed by means of petri dishes. Lots of glassware, petri dishes are small boxes of glass, in which you have a sort of a nutrient medium and into which your gene of interest of antibiotic resistance is put, and it is just packed back to back from one part of the wall to the other part of the wall without any sort of a gap, and thereby you are getting multiple copies of the gene of interest in your sort of a Petri plates. If you have a Petri plate, you have a gene of interest and all of these are aligned. They are just packed just like as you pack matchboxes or you pack Petri plates compactly in a sort of a closed room. You can have millions and millions of these test tubes or glasswares with the gene of interest which are just fully loaded, and you will have your sort of gene of interest, and this technique is termed as gene cloning. So, what exactly is the goal of biotechnology or genetic engineering? One of the goals of genetic engineering or biotechnology is gene cloning, that is making multiple copies which of exponentially and geometrically increase in the number of the genes of the desirable product for human or mankind, primarily for use for medicine, for industry, for agriculture, for again for environment, as well as for other sort of things. So, we are coming to the broad scale application, large scale application of biotechnology. If ever you want to make a novel protein, if ever you want to make a new drug, if ever we want to discover a sort of a drug to fight malaria, if ever we want to discover a sort of a drug for all the sort of a new diseases or all the antigens and the antibodies and the vaccines for all the new diseases which can suddenly crop up in the 21st century due to climatic change. And if ever we want to develop novel proteins, novel enzymes, novel drugs, novel foods, the only answer is biotechnology. Conventional wisdom and modern technology should take the sort of is the blend for biotechnology. So, the take home message for students of biotechnology when at the end of the sort of a module is, we had started biotechnology stating how it is a sort of a science, it is an interdisciplinary science. How we had seen what is the exact meaning of old biotechnology is fermentation biotechnology. It was microbial based process. Then we had come into the sort of a definition of the EFB definition of biotechnology, which says we are going to go for large scale for human welfare, and it is an integration of both old science as well as new science. Then we stayed out with relation to understanding that if ever we want to learn more about biotechnology, it is premised on two important conceptual backgrounds. The conceptual background is principles of biotechnology and plant tissue culture. Then later on we understand that biotechnology is an integral part of plant breeding, though plant breeding in the conventional plant breeding and sexual reproduction and asexual reproduction, there can be a mix of desirable and undesirable characters, whereas in modern biotechnology it is precision biotechnology and we can locate your sort of a gene of interest. Once you locate the gene of interest should be manipulated, it should be modified, it should be cultivated, it should be generatable, and you should be able to make large scale copies. And if you want to make large scale copies, we should find out what is the method. And the only method which we got from the breakthrough science of Boyer and Cohen is how do you develop a sort of a recombinant DNA molecule. So once you know how to develop a sort of a recombinant DNA molecule, with the recombinant DNA molecule, the next step is from recombinant DNA molecule to gene cloning. That is making multiple copies of the particular sort of gene. So thereby the flow charts primarily says identify the gene of interest, introduce it into host cells, and later on maintain it in the host cells and transfer to the progeny of E. coli. These are some of the basic first steps in genetic modification. So when it comes to the working of biotechnology, the term which is used that is termed as genetic modification or genetic transformation. So genetic modification or genetic transformation applies to host cells which have got a sort of a recombinant DNA, and multiplication of your gene of interest into large numbers, into large copies of your gene of interest is termed as gene cloning, and the process is termed as biotechnology, and it is based on basically some principles, and in the principles you've got genetic engineering and plant tissue culture.